Okay. All right. Well, thanks for for joining and participating, and thank you to everyone who's watching. This is part 2 of a debate that focuses on the motion evolution and abiogenesis provide the best explanation for the existence and diversity of life. And because of the broadness of this topic, we ended up splitting it up into its 2 fundamental constituents, where in the part 1 of the debate, we focused on abiogenesis. And, just for anyone who didn't see it, in the previous debate, I basically made the argument that abiogenesis is the best explanation for the existence of life in the first place, drawing from aspects of the real world that we know to be real. Basically making the case that it's the although there's a lot of details we still don't know about the history of life, it's the only explanation that invokes phenomena that we can confirm to be part of the real world. Drawing from for example, the things that are required for abiogenesis to occur are things that actually happen in the real world, like in the world of chemistry, for example, like organic synthesis and molecular self-assembly. My opponent made some objections that, made a number of objections that, to the best of my understanding, I don't want to put words in your mouth, feel free to correct me if I get any of this wrong, but they tended to follow the theme of saying that there's a lot we still don't know. We haven't been able, for example, to resurrect, the primordial living systems in under laboratory conditions, and so there's a number of unanswered questions about this topic. And so, in light of all this, we should be more humble and admit that we don't know, or to say that we don't really have an explanation necessarily. And to a certain extent, I think that's pretty fair. This might come down to a difference that we had over what it means for something to be the best explanation. Granted, there's a lot we still don't know. That's why it's an active area of research in the first place. In any case, the discussion of evolution assumes that life already exists, irrespective of how it came into existence. And this topic, or this debate, focuses on the motion that evolution is the best explanation for the diversity of life. And I argue that evolution is the best explanation for the diversity of life because of common ancestry, basically. The statement that evolution is the best explanation for the diversity of life is the logical conclusion from 2 premises. The first premise being that all life on Earth, as diverse as it is, descended from a common ancestor, and it acquired this degree of diversity as it descended through the process of descent, leading back to a common ancestor. So, if we could hypothetically rewind the clock and go back to the original origin of life, we would see that life becomes progressively less diverse. And so the first premise you could summarize by saying that that life comes about by a common ancestor, and that's how it gets its diversity. And then the second premise is that common ancestry, descent of all life on Earth today from a common ancestor, is encapsulated within the theory of evolution. It's part of the theory, and therefore it follows logically that evolution explains the diversity of life. So a lot of conversations and debates about the subject of evolution tend to focus on various aspects of what evolution can or cannot do, focusing on things like, evolution can't, for example, build an eye or a bacterial flagellum, different living systems that the challenge is put forth. How does evolution explain this? And I think those are fascinating topics. I love talking about them. But I wanted to do something a little bit more ambitious because we can find individual instances of systems that evolve by the cyclic iterative process of mutation and selection, but a lot of focus is taken off of the topic of common ancestry, and so I'd like to describe some of the evidence for common ancestry.
Yeah, I'd like to hear it.
Yeah, sure. Okay.
Maybe let's can you present maybe evidence points one by one, and then maybe we can chew them up, digest them one at a time instead, and if they need, because each one may merit debate.
Right. Yeah, for sure. Okay. Yeah, no problem. So, well, I would say in a nutshell, the best evidence for common ancestry comes from phylogenetics, and in particular, comparative methods in phylogenetics. And so this basically comes down to various methods by which scientists study relationships between living organisms and use this evidence to retrace the history of life. And so when I say comparative methods in phylogenetics, what I mean is that you can use different bodies of evidence collected using independent methods from different fields of science, and they converge on the same answer as far as which organisms are closely related or more distantly related from which other organisms. And this is the line of reasoning that scientists use to reconstruct phylogenetic trees. You know, the evolutionary tree of life that you might see in a like a biology textbook.
So can I ask a question? Can I clarify?
Sure.
Ask a question to clarify. So, okay, so is phylogenetics, in simpler terms, is it simply looking at a genome of this species versus this species, comparing them and see how similar they are, and then you, based on the similarity, you can deduce how far close they were in, you know, the ancestral line from each other, or from from their least common ancestor?
Yeah, that's you're on the right track. There are some differences. Phylogenetics is not first and foremost. It's not limited to comparing genomes. There is, you know, phylogenomics, which is, you know, the application of phylogenetics to whole genomes. There's also, of course, you know, phylogenetics based on other lines of evidence. There's, you know, fossil phylogenies and phylogenies built from biodiversity and protein biochemistry and things like that. So when I say comparative methods in phylogenetics, what I mean is that there's different independent lines of evidence that converge on the same tree, the same branching topology for the history of life. So if I said that, for example, humans are more closely related to chimpanzees than we are to any other species of primate, but we're more closely related to any other species of primate than we are, for example, to ungulates. And
Sorry,
I don't want to talk while you're distracted.
you're good. You're good.
Okay, so if I said that, you know, humans are more closely related to chimpanzees than any other species of primate, but we're more closely related to any other species of primate than we are to other placental mammals, but we're more closely related to other placental mammals than we are to marsupials, and you can back it on, you know, you can zoom wider and wider out on the tree of life and do the same sort of statement for when it comes to, you know, birds and reptiles and amphibians and fish and invertebrates and animals. And by non-animals, of course, I'm kind of lumping all of this together, you know, fungi and plants and protists and bacteria and so on. So this is a particular specific branching topology that's not arbitrary. And so the confirmation of the common ancestry of life is the fact that we arrive at this same conclusion, this same branching topology for the tree of life, using completely different independent bodies of evidence collected and analyzed using different independent methods.
What are, and then can you give me like three of the examples of three of those methods?
Yeah, sure. So one example is, let me unpack it in the following way. All animals are what we would call multicellular heterotrophic eukaryotes. You know, meaning they derive their energy from the consumption of organic material rather than photosynthesis, and they're multicellular. And of all the animals that fit this you know, these criteria for classifying it as an animal, some of them have a dorsal nerve cord and some of them don't. We call those that do, we call them chordates. And of all the animals that have a dorsal nerve cord, some of them have a backbone and some of them don't. And of all the animals that have a dorsal nerve cord, a backbone, some of them live on land and are what we would call terrestrial tetrapods, you know, having four appendages, hence the term tetrapod. And some aren't, and those that aren't, we tend to classify as you know, fish. Although fish is a huge umbrella term. There's more species of fish than there are amphibians, you know, reptiles, birds, and mammals combined. So, of course, I'm kind of oversimplifying for the sake of time. But of all the terrestrial tetrapods, you know, some are warm-blooded and some aren't. And of all those that are warm-blooded, some have fur and some don't. You know, those that don't, for example, in particular, birds are warm-blooded and they don't have fur; they have feathers. And of all the animals that have fur, you know, you can make similar statements and continue to constrain the part of the tree of life that you're looking at by adding on more and more criteria. And as you do that, you see fewer and fewer animals meet these criteria. And so the classification gets narrower and narrower until we arrive at any one species. You know, it can be human, or it can be, you know, chimpanzee, or it can be, you know, rats or mice or whatever. Mm-hmm. And the thing that's worth noting here is that these are not just arbitrary characteristics. These aren't like injuries or scars. These are derived characteristics that we inherit from our parents. Every single characteristic I mentioned is an acquired, inherited characteristic, and so it's encoded in the DNA. And so if you change the DNA of a fertilized egg, you can hypothetically change, not hypothetically, you can actually change what animal the fertilized egg develops into. So, another way to look at evolution is to say that it's every generation, it's just one cell, one fertilized egg developing into an animal. And then over the process of reproduction, some new genetic mutations occur, and it makes some slight modifications to the animal that fertilized egg develops into. And on a short time scale, you don't see a lot of change, although in some instances you can, like with, for example, selective breeding. Cats, dogs, roses. There's animals used in agriculture, but in any case, it's these are derived characteristics that map onto a what we would call a nested hierarchy. That's what I mean when I say that these are combinations of character traits.
What kind of hierarchy?
I'm sorry?
What was that?
A nested hierarchy. So it's a group of organisms, nested within a broader group of organisms that's a little bit less specific, and then nested within another broader group of organisms.
Okay, I mean, that makes sense. That makes sense as far as like organizing animals according to the characteristics. And yes, some are gonna, so they begin to branch out, like you said, four-legged animals, and you can divide those in their own categories, so on and so forth. And that's known as, what'd you call that? What's that method known as?
I'm not sure which aspect you're referring to. You mean the nested hierarchy?
Sorry. So going back, not nested hierarchy. Well, is that called the nested hierarchy? What you were just describing, as far as, because I was asking for 3 methods of the of like phylogenetic.
Right. So this is a nested hierarchy that specifically refers to organismal traits. Traits that you can see, traits that Darwin would have noticed if he were studying these animals.
Okay.
These are not traits that you need to do modern molecular sequencing or look at a cell under a microscope in order to notice these traits. So these are I'm sorry.
But that, are you saying that what you're describing, like the idea of physical characteristics and the nested hierarchies associated with those, that doesn't necessarily, does that support common ancestry by itself, or is that, along with something else, helps support the case for common ancestry? Like, what I'm saying? Right. So if I'm organizing all the animals and creatures in the world, what's going to lead me to make the jump to saying, hey, they must have all come from the, from a bacteria?
Right. So,
something as simple as that.
Right. Yeah, exactly. That's good. Going back real quick to the nested hierarchy of these, and you can make a similar nested hierarchy in reference to bacteria, or in reference to plants or fungi. I'm kind of sticking to animals for the sake of simplicity, where most people are more familiar with animals. But in any case, the nested hierarchy is something that as you zoom in closer on any one branch of the tree, like take for example, any subclassification of animal you want, really, it's all the same. The issue is that you have an ancestor that leads to descendants with increasing diversity. This is something that on a relatively short time scale we can see. And so the question is, how do you get a diversity of traits that are all nested within a broader category of traits, nested within a broader category of traits? How if you want to ask the question, how what kind of phenomena can produce this kind of effect? Well, we have a phenomenon that we know of and that we can point to, and that is diversification from a common ancestor, and we can see examples of it, like for example, selective breeding in dogs. Now, I understand selective breeding is something that humans do interventionally, but the principle is pretty similar. You don't even have to focus on dogs, for example. You can take roses, or any organism that's been selectively bred. For agricultural purposes, for example. If you've ever seen a Princess Anne rose, they're huge, they're beautiful, they're colorful. They don't look anything like a wild rose. Same thing, in terms of various breeds of dogs. Mm-hmm. Those are easy go-to examples. But sorry, real quick. I promise I'm going somewhere with this.
That's good. I'm enjoying it. Go ahead.
Okay, cool. So the thing that Darwin noticed is, or I shouldn't say that he noticed, not that he was the first one to notice. This is something that this was an area of, this was a set of emergent conclusions that several scientists in the 1800s were gradually coming to. So, he was the one that was most famous for coming up with the idea of natural selection, but he wasn't the only one who did. But in any case, the thing that they noticed in the mid 1800s was that other selective breeding is something that humans don't have to do interventionally. So for example, if you look at a predator-prey relationship, it's very real to say that in a sense, antelopes are selecting for faster and faster cheetahs, and cheetahs are selecting for antelopes that are progressively better and better at getting away from the cheetahs are selecting for antelopes that are better and better, gradually better at getting away from the cheetahs. And by selective breeding, I mean that it's the antelope that gets away from the cheetah that is more likely to pass its genes into the next generation. And you could say same thing for the cheetah. It's the cheetah that doesn't succumb to starvation, that's able to actually catch the prey, that is able to more successfully pass its genes into the next generation. Although the trait doesn't have to be predator prey, it doesn't have to result in death. It can just be minor differences in reproductive success. So for example, insects are selectively breeding flowers, for their nectar and for their color, in this very same way that humans are selectively breeding flowers. It's just that insects aren't doing it with the same sort of cognitive intent that humans are, but they're still selectively breeding for flowers. And flowers in turn are selectively breeding for insects by providing them with nectar and helping insects survive and reproduce. And so we have at least a system of observations we can make about life in the world today that produces similar effects as what we see for all of life. Now, this doesn't prove that all life has a common ancestor, but it provides at least a plausible hypothesis that we can go out and test by trying to see if more information matches that hypothesis.
Okay.
And that's where other lines of evidence come into play. So for example, I think the best confirmation for common ancestry is, let's take a look at an evolutionary tree that was created before we had access to other lines of evidence, and then see, does it predict the same evolutionary tree once we do have those other lines of evidence? For example, if you were to open up a 1936 paleontology textbook, the 1936 textbook on vertebrate paleontology by Alfred Romer, for example, there's a number of phylogenetic trees for animals, for vertebrates at different scales. So like there's a primate phylogeny, and then on a different page there's a phylogeny for placental animals, and on another page there's a phylogeny for that shows where where mammals are in relation to reptiles and amphibians and so on. And this evolutionary tree, this phylogenetic tree, makes a very specific prediction, because the number of trees that the number of branching topologies that a tree can have increases exponentially with the number of branches that you add. So for example, a tree that has only two species and one common ancestor, there's only one way to draw the tree. But if you've got hypothetically four different species with one common ancestor, well, now there's over a dozen different ways you could hypothetically draw that tree to show the four descendants coming from that one ancestor. And it increases exponentially, so that by the time you get to a couple dozen species, now there's we're talking about in the millions of number of different ways that tree can have a specific branching topology. And so if you have two lines of evidence that independently map onto the exact same branching topology, we're talking this is a confirmation of a specific version of the history of life that defies millions to one odds. And that's why scientists can statistically say without lying that, we've confirmed that this species is such and such closely related to this other species, to the tune of 99.999% confidence, or somewhere in that range. I don't want to hog all the conversation. I'm curious to know what your thoughts are.
I mean, I was with you on like some of the nested hierarchy. It kind of got lost a little bit on the the second thing that kind of backs it up. Here is, I guess, here's a topic I want to camp on, I guess. I mean, phylogenetics, it sounds good. What here's a question maybe that can take us in a more dialogic direction. What like observational laboratory evidence do we have that shows evolution happening, like developmental evolution. You mentioned selective breeding in dogs.
Mhm.
And so here's what I want to kind of address and not camp on too long, but so selective breeding in dogs. Yeah, we do selective breeding, right? We've got all these different breeds, everything from the Chihuahua to the, Great Pyrenees, those massive, white mountain dogs. But every time we're selectively breeding, aren't we creating a bottleneck in every specific dog breed, especially the ones that have been inbred to preserve like some very, very specific characteristics, are left with a less robust genome than their ancestors. Am I correct in that?
Yeah, so there is a danger with population bottlenecks. You're absolutely right. Yeah, sorry, I thought you were done. Feel free.
Okay, so here's a follow-up question. From what I understand, like bottleneck, for the most part, is not what you want to have as a developing species. But in what cases have we seen an organism go through a specific species go through a bottleneck, and in resulting generations have been left with an increasing or more robust genome?
Well, generally it doesn't happen. At least it doesn't happen that quickly or immediately. There is a problem that presents itself when you have a species that is, passed through a genetic bottleneck. Just out of curiosity, I don't want to lose anybody who's watching, but I also don't want to give you information you already know. So, do you know why bottlenecks are dangerous and why, for example, inbreeding is bad?
Yes, because it's the different, the variation of alleles gets severely reduced, and the mutated genes that have been built up are more likely to become dominant and cause problems.
Well, it's not necessarily that mutated genes are more likely to become dominant. What happens is that you've got when a mutation,
less variation results in the less ability to adapt to environmental factors that may change.
Right, but the reason for that, the reason why that is the case is because you have recessive mutations. Well, generally speaking, it's because of recessive mutations that are lurking in the genome that don't present themselves until, two organisms, just by just by stochastic effects, by the random the law of independent assortment, law of genetics. Two organisms that just happen to be recessive carriers of the same mutation or of a deleterious mutation in the same gene are more likely to produce an offspring, a descendant that just by chance inherited both defunct or dysfunctional copies of the same gene. The likelihood of two parents running into each other that both have a mutation in the same gene increases the smaller the population is, or the more inbreeding that there is. And so yeah, you want ideally in order to protect against the dangers of inbreeding and population bottlenecks, then yeah, you do want increased genetic diversity. You want if a mutation happens, you want it to be diluted through the gene pool so that you're not at increased danger of running into a mate that's going to have, just by chance, the same recessive mutations that you have, so that your kids are more likely to develop genetic abnormalities. So yeah, all of that is to say that yes, that is the case, that you want that population bottlenecks can be dangerous for the survival of a gene pool. However, you don't necessarily need population bottlenecks in order to get evolution going. Or at least not dangerously constrictive.
But isn't every branch, every fork on the tree of life, doesn't that denote a bottleneck happening? We have
well, it depends on where we set the bar for what constitutes a genetic bottleneck. I mean, a population shrinking in size because it becomes reproductively isolated from a sister population is not necessarily the same thing as going through a bottleneck, as long as the population is still relatively large enough to overcome the effects, so to speak. It's not, part of the problem is that it's not a hard and fast rule. There's a continuum of, as the population gets smaller, the chances of the negative effects of inbreeding increase, and there's no specific line where you hit that, okay, now it's a population bottleneck. So the idea is that as long as you have a large enough gene pool to dilute the effects of negative mutations, such that the population, or that the gene pool doesn't go extinct, then evolution can still proceed forward.
Okay, yeah, I'm tracking with you on that. So from what we've observed, every time you're, even from what we know from, breeding new dogs to get dogs that look, completely different.
Mm-hmm.
We know in those cases that you're still left with a less robust, genome, with a less, robust, or a less variable, booklet to draw from. So has there ever been an observed situation where, we see DNA increasing in a productive manner that's, generating either new systems or maybe even new traits, that shows us that, okay, evolution can take an existing genome and work on it, upgrade it into a more complicated or complex, creature. As far as, like, I guess bacteria would be a good look at that.
Yeah. So, excuse me. So I'm not sure exactly which point you want me to focus on, but as far as, can mutations add something adaptively beneficial to a genome? Is that roughly equivalent to what you're asking?
You know what? Yeah, that kind of was, covering a lot of ground there. Here's what I'll point it to. How does a bacteria become a beetle? Have we seen what scientific evidence do we have, live observation type stuff that shows us that a genome is malleable, flexible, novel, innovative enough to develop in that direction?
Right. So that goes back to something I said earlier, where a fertilized egg develops into, an adult of whatever species it is, depending on what's in the biological molecules that fertilized egg inherits from the parents. Mm-hmm. So there's biomolecules, there's proteins, there's DNA in the sperm, there's proteins and DNA in the egg before it becomes fertilized, and then the DNA and the proteins in the I say that the DNA because there's a distinction between genetics and epigenetics, of course. So it's not just the DNA sequences that are doing the work; it's also the regulation and the function of the genes that are doing the work. But in any case, for simplicity's sake, I'll say the DNA. If you change the DNA of the fertilized egg, you're going to change what animal it develops into. And when it comes to something like different features in different animals, for example, generally what happens is that I don't even know if generally is fair. What happens is that over the course of embryonic development, you have this phenomenon called asymmetric cell division, where some cells divide a little bit more faster than others. Some cells will turn certain genes off, other cells will turn certain genes on. So for example, every cell in your body has identical copies of all the same DNA, but not all of them are turned on. The genes for making a neuron are turned on in your neurons, in your neuronal cells. The genes for making, a muscle cell are turned on in your muscle cells. And the genes for making a neuron that are not required for muscle cells are not turned on in your muscle cells. And so there's this phenomenon of the activity of the genes being turned on, turned off, the differential regulation of the genes leading to certain cells growing, a little bit more faster than others, a little, dividing at a higher rate than others. And that leads to, elongation of certain traits or truncation of certain traits. And that's how you get, features that differ between species. You asked specifically, a beetle from a bacteria. I don't know if that was like a one-off example you wanted me to speak to, or if it was the specific thing you wanted me to focus on.
Just a general principle of, like, okay, if we're starting as bacteria, a bacteria from a beetle, I understand, is a huge jump. And so I know that that's not going to happen, overnight or even a few million years. But you still have to, we can look at bacteria DNA. And what I'm thinking is, what comes to mind is the Lenski experiment. 68,000 generations of E. coli bacteria, and the end result is E. coli bacteria. And so, which is the equivalent of 1 million years of human generations. And from what I understand, the current number of how long it took us to, descend from an ape-like ancestor was like 12 million years for humans to, come into our own vogue. But if after 1 million years we haven't seen the E. coli bacteria make any significant modifications except the ability to digest citrate, which already was part of its existing genetic code, then why are we assuming that it can make a huge jump to possibly turn into any other animal within a few billion years?
Yeah, so it's not a huge jump. It's a series of little jumps, and if you want to. If you want to bring it back to like the common ancestry, for example, of E. coli or any species of bacteria and say beetles or any species of animal, a simple exercise to at least help wrap your mind around the problem is to look at what are organisms that have some sort of, well, I hesitate to use the word transitional because it's not that an E. coli evolved into a beetle. It's not quite how evolution works. It's that E. coli and beetles share a common ancestor, but something that might help elucidate how plausible it is would be to take a look at what various transitional species along those two different trajectories would look like. And so, for example, we have in the fossil record simpler analogs to beetles. We have things that as you go further back in time, there are multicellular organisms that are not as conducive to fossilization or that don't have as hard of an exoskeleton, but they still have some features in common with beetles. There's small little primordial arthropods, for example, that have the ruminants of modern arthropods, but don't have all the same features. Like I'm blanking on the name, but
okay.
It looks like you want to say something.
Yeah. So, but here's the issue. Here's the problem for me, I guess. So as man, skeptical of kind of thought exercises because you can look at the different patterns, just patterns of correlation of similar characteristics and so on and so forth, and keep tracing it back. But I'm still skeptical that the common ancestry is more of an assumption that is backed by a lot of thought exercises because
let's not do thought experiments then. Let's look at the evidence because common ancestry is a testable hypothesis that's confirmed by the evidence.
Okay.
And that's what I'd love to focus on. Actually, it's what I prefer talking about. So
okay.
Okay.
Yeah, go ahead.
Okay. Yeah. So you mentioned a few minutes ago, or you asked a question a few minutes ago that elicited from me a response that involved talking about the nested hierarchy of traits, and then that led us into talking about
before you go on, do you have evidence of like kind of observable live action experimentation? Is that
So first, I think one way to do it is to start by saying that start by laying out a proof of principle. We know for independent reasons that you can do genomic comparisons of genomic data in order to retrace history. And we can do this in an accurate way. For example, if you gave me the DNA in unlabeled test tubes, didn't tell me which DNA came from which person, but you gave me the DNA in unlabeled test tubes for every member of your extended family, all your cousins, grandparents, aunts, uncles. I could do the sequencing of the DNA and I could reconstruct your family tree with 100% accuracy. And I would be able to, I wouldn't know necessarily the names of the people, but if you pointed to where you were on the tree, I could say, okay, well, these 3 DNA samples are your brothers and sisters, and then these ones are your cousins, and so on. And we can do this with other data sets that don't even involve humans. For example, there was an experiment in the early 90s that was published, I believe, in the journal Science or Nature. I'll post the link below in the comments section. But what they did was they took bacteriophages, viruses, and they generated an experimental phylogeny where they took one population and split it, and then they split those 2 different subpopulations, and they propagated these populations to generate an artificial phylogenetic tree. And then a different group of scientists, completely blindly to the original experiment, just took the genetic data and they were able to reconstruct the phylogenetic tree with the correct branching topology. And we can do this, for example, with, do you mind? Sorry, I promise this is related, but if you don't mind me asking a personal question, do you consume alcohol?
Not really.
Okay, that's fine. I'm a bit of a beer drinker, not all the time, but in any case,
Kind of sacrilege out here because I'm from Colorado, which is like almost brewing headquarters.
Right. Yeah. Well, I'm originally from Oregon. There's a booming wine industry.
Okay.
But in any case, one thing is that there's a huge variety of different types of alcohol. There's wine, yeast. There's wine. There's beer. There's different liquors, and they all use yeast for fermenting the alcohol, or fermenting the fermentation is the conversion of alcohol to sugar. And over the course of the history of alcohol agriculture, yeast, just like animals or plants, have been subjected to domestication, and there's different populations of yeast that are they're all yeast, but yeast is really diverse. There are species of yeast that are more closely genetically related to humans than they are to other species of yeast, for example. It's an incredibly diverse array of yeast, but incredibly diverse group of organisms that we're calling yeast. In any case, when it comes to the alcohol industry, actually, you can use genetic sequencing data from different species of yeast, or not. I don't know if species is the right word. I'm not aware of whether they've done like hybridization or experiments to determine whether they've speciated. But in any case, there's different strains of yeast. There are some that are used for beer that are not used for wine, and there are some that are vice versa. And what they can do is they can actually reconstruct the entire history of the alcohol industry going back to ancient times using just the genetic data from modern yeast. And the same thing, it's the same principle as when I mentioned earlier, where we can reconstruct your family tree using the DNA of everybody, and we can reconstruct
you could do the same thing with every dog and know that they trace all their ancestries back to a pair of ancestral wolf-like animals.
Right. So when we do the exact same thing for different species that are as diverse as, say, beetles and bacteria and chimpanzees and humans, what we get is not only a family tree that's arbitrary, not only a family tree, because that could be arbitrary, but we get a tree that has the exact same branching topology as completely independent lines of evidence, like the one in the 1930s paleontology textbook. And the reason I brought that textbook up is because that was a phylogenetic tree that was elucidated using the fossil data back in the 1930s, which was before we had DNA sequencing technology. So this was a completely independent line of evidence that we didn't know what DNA sequences would look like at the time. We didn't even know that DNA was the molecule of inheritance that was discovered in the 1950s.
So was there ever any cases where two creatures that looked similar were actually a lot more separate, on the genetic level, that had to get moved over a species or reclassified?
Well, in very rare cases, in very minor cases, yeah, that does sometimes happen, and there are reasons for that. We can account for that.
And the reason I ask, because if that's the case, then I guess it would make sense that if humans and primates share a lot of characteristics, so I would assume that genetically they'd be a lot more similar.
Yeah, but there are some. I'm sorry, I don't mean to interrupt you. Go ahead.
And so that's the point. It's like, well, it's just backing up what you can already kind of deduce, but you can already deduce from just observation. However, it doesn't necessarily follow that everything evolved from a lower life form into what it now is.
But it doesn't. It doesn't just. It's not just circular or self-confirmatory of the other lines of evidence, though, because in a number of different cases around the genome, there are loci that are shared across species and that perform the exact same function in different species. So, in order for the genetic data to be circular, what would need to be the case is that all the genetic data we use is it corresponds, or differences in genetic data correspond to differences in organismal biology, and that's not necessarily the case. In fact, we can reconstruct the same phylogenetic tree and get the same result, the same conclusion, when we use genomic loci that are completely independent of organismal biology, completely independent of animals, the specific organismal differences between different species of animal, or between animals and non-animals, for example, between animals and plants. A good example is ribosomes. The ribosome performs the exact same function in every species on the planet, from bacteria to humans. And the reason we know this is because you can take a human gene and clone it into a bacterial cell, and the bacterial ribosome manufactures the human protein by reading the human gene just perfectly. This is actually how we make insulin in the pharmaceutical industry on an industrial scale, is with genetically engineered bacteria cells that are synthesizing human insulin. And so we know the ribosomal genes perform the exact same function no matter what species they're in. And yet, when we use the DNA sequencing data from ribosomal RNA genes, it corresponds to the exact same tree that you find in independent lines of data, like the organismal biology that I mentioned when I was talking about, some animals have a backbone and some don't, and all that. When I went through all the list of different shared genetic traits, you get that same basic nested hierarchy. There are some differences, and we know that there are some exceptions. There are some instances, just like you said, where we say, okay, we thought that this species was more closely related to this one, and it's actually more closely related to this other one. And when that happens, it's generally because of, well, for one thing, it's generally because there are quite oftentimes very small data for doing molecular phylogenetics, and there's things that can happen like homoplasies, where the same mutation occurs independently in two different lineages, and it makes the organisms look artificially more closely related than they actually are if you're using a really small data set. But if you use a much larger data set like whole genome sequencing, then you can actually resolve the incongruencies between different phylogenetic trees, and you get the exact same tree consistently.
What was what'd you call that, of the descending of like the ribosomal DNA?
I'm sorry, the descending of ribosomal DNA?
Was there a specific study or was there a specific thing that's called, like, as far as that, tracing that back, what that study, what that metric is known as?
Yeah, so there's one, well, it's molecular phylogenetics. So at the beginning of the talk, when I said the best evidence for evolution, for common ancestry specifically, is comparative methods in phylogenetics. One of the methods is molecular phylogenetics using DNA sequences. And the specific, there were a couple of papers in the early to mid 90s. There was one in the journal Molecular Biology, a couple of them in the journal Molecular Biology and Evolution. And then there was another study in FASEB, which is the name of another journal, and yeah, they collected the sequences for ribosomal RNA genes, and this is a set of genes that are commonly used in evolutionary studies, because the genes have the same function across all different species we've ever studied. And so it provides a good control for just making sure that we're not using genes that are redundant to the organismal characteristics that we were using for constructing a different tree.
So all these studies, they show that, that we're all not just related to each other, but we actually, I feel like all the studies are showing that we're related to each other in a way, similarly with, ribosome and genetically, so on and so forth. But I guess I'm not seeing the connection between how we're all descended from, the same common ancestor.
Right. So that's revealed in the phylogenetic tree. There's different. So the one thing I mentioned at the beginning, and we're running out of time. I'll give you the last 30 seconds or so if you'd like, and maybe we can do part 3 if you'd like. But in any case, phylogenetics is a method of retracing the history of life using the specific distribution of shared and unique traits, and it's not just as simple as saying these organisms are more similar, therefore they're more closely related. You're also looking at the specifics of the distribution of differences. In terms of genetic phylogenies, it's the distribution of genetic differences in the terms of DNA sequence. In any case, different independent methods for retracing the same tree converge on the same answer, the same branching topology, and that's the evidence. That's the confirmation. Sorry, I know that's less than 30 seconds. Go ahead.
You know what? Yeah, let's definitely try to, you know, do another dialogue on this, because there's, man, I've got a bunch of other questions and, you know, arguments and that I think are worth talking. So.